Shall I get going? Okay, I think we'll get started as we're a little bit behind time already, but by a few minutes. And it is the last session, so we don't want to, start too late and keep you all from the snacks later. Okay, great. So the next session that we're going to be diving into is about creating robust ecosystems for microbiome research in Africa. So yeah, it's get going. Wait, we skipped a slide. There we go. Okay, this is me. I'm Nicki Tiffin. I work at the University of the Western Cape. I'm at the other SANBI, the South African National Bioinformatics Institute. And it is confusing because there's a Biodiversity Institute, but I just want to say we were there first. So yeah, we're going to be talking about robust ecosystems for microbiome research. You're going to see me squinting a bit because I left my specs in the car. And for everyone who arrived by car, you know that the car is very far away. So also when we get to the questions, you'll have to say who you are, because even if I know you very well, I won't be able to tell from here if you're sitting anywhere from the middle of the room back. So I apologize for squinting and peering at my slides. So I'm going to talk very briefly before we bring our fantastic speakers up here about our sort of views of an equitable environment for microbiome research. And I thought I would start by talking about our aspirations for equity, our equal partnerships by design. This is where we really want to arrive. So we're talking about equal relationships between partners, and this sets by having equal relationships, we set the foundations for equitable health collaborations. We want to have African populations that can benefit from African research alongside global populations. It's not about excluding anyone else. It's about including the Africans. And we want African researchers to be the intellectual owners of the research that they're doing and the work that they're generating on the continent. And then, yeah, African research ownership can support local capacity development and ensuring that we advance what's happening on the continent as we go. So our aspiration is really for African sort of generation, the Africans being the generators and the custodians of the African data and biospecimens. This is what we're aiming for. Of course, equity in practice looks quite different, and we have really historical, global, and systemic inequity in the way things happen. So, yeah, we have ongoing and historical expatriation of the resources that we develop here. The samples and the data generally get shipped off continent, and that's where the rest of the stuff happens. And then we have kind of with this unidirectional flow of content, it's often pushed actually by funders and by journals that insist that you send your data and sometimes even your biospecimens to centralized repositories where they can be more generally accessed. So this is why I call it systemic. This is not like an accident of some people expecting this. This is a very fundamental demand by many of the most powerful institutions. And this really pushes a unidirectional flow. And then we also have, the very well kind of resourced global North partners who can often sort of take over the analysis, take over the publications, the data analysis, the storage, and the work that follows on often ends up being pushed also to global North centers. And frankly, the African researchers are often not resourced enough to compete for those positions. They're not resourced enough to compete for senior or corresponding authorships. They're not resourced enough to insist on keeping the analysis locally. So these are really quite serious problems. And we have, again, a systematic undermining of African sort of resource generators, the researchers, from participating and owning and benefiting from their own research in their own countries and their own populations. So what does an equitable research ecosystem look like? Data and biospecimens are stored where they originated. They don't actually need to be expatriated somewhere else. Researchers who generated the resources and work with the local populations do the research locally, and they have collaborators who participate and contribute. And this, again, is a very important point, and nobody wants to close the doors on our wonderful collaborations that we have, but we want to be in a driving seat for the collaborations that involve our populations, our people, where we're doing the work on the ground, building the community engagement processes, and winning the trust of local populations. And importantly, also understanding local context. Yeah, and our participants really also need to be part of this process. And if you're sitting off continent, it's very hard to make that happen. Yeah. So these are just some of the underpinnings: participant-centric research, resource sharing agreements, benefit sharing, which seldom features actually in a formalized way, African leadership, African ownership and analysis, African research storage, and African capacity development. And we really want our international collaborators to respect this, so it shouldn't really be a fight. And of course, there are the legal and ethical requirements that need to be upheld in these processes. And often when we're working on the ground with these things, this is what we can drive. So I can see I'm running out of time very fast. I'll just flick through very quickly to say that we can make a lot of this happen by using more creative ways of sharing, by not thinking only of unidirectional flows, but more collaborative approaches. And we've built various tools in my group that I've just put some links there, but you can also contact me if you're interested, that help us do more equitable research. So they're very practical tools that you can harness to move beyond this unidirectional shift of, I have these things here in Africa and I give them to you elsewhere to do what you will with. Just to mention, finally, it's a quick plug for a project that my group is doing, funded by the Bill and Melinda Gates Foundation. We're building a platform that aims to level the playing fields for the reuse of data and biospecimens generated here on the continent. The idea of the exchange is that if I'm holding resources, data or biospecimens, I can advertise them on the exchange, and interested parties can make contact with me through regulated approaches on the platform. We can negotiate how we're going to do our sharing, build an MOU or an agreement, and sign it off before anything has to be handed over by me as a generator, so that I can be confident that whatever is happening next with those resources is something that is legal, it's ethical, it's fair to us as Africans, the participants who have contributed, the researchers, to take it forward. And this will be online by the end of the year, so keep an eye out for it. Great. So we've got a great session coming up. We've got 3 excellent speakers. So Ovokeraye is online. So if I can just introduce Dr. Ovokeraye Oduaran, who's a leading expert in the microbiome field with a focus on the gut microbiomes of diverse African populations. Together with the Awujane Collaborative Microbiome Group, her researchers uncovered novel microbial taxa and biomarkers linked to lifestyle transitions. She's passionate about global health and a thought leader on equitable partnerships in microbiome research in Africa, leading the H3 Africa Microbiome Task Force and the African Microbiome Special Interest Group in the organization of the African Microbiome Day event to showcase microbiome research on the African continent since 2022. And I'm going to say microbiome in my sleep many times tonight by the end of this. So yeah, over to Dr. Oduaran, who's online. So just keeping an eye if we've successfully made that transition. I can see her there. Okay, we're just getting onto it. There you are. Yes.
Great. Hi. Can you see my screen properly? Do you see the presentation?
Done.
Okay, great. So, fantastic. Good. Oh, I don't see my screen. 1 second. That's okay. Okay. All right. I hope I'm able to navigate this. Because for some reason I don't really see my screen properly here. That's okay. Just to say thank you very much. Good evening, everyone, and my apologies for not being there in person. The discussions have been fascinating so far. The ones I've been able to get to catch. Just to say a big thank you to John and the entire team for the invitation. And as Nicki says, I will be talking a bit about just go back here for a second. Sorry, I'm struggling with my slides a bit. Rethinking microbiome research in Africa, unpacking the 5 pillars for more equitable partnerships. I'll start by just giving a bit of an outline of what I plan on going through today. Start with the context of this comment, which Joanne featured. Thank you so much, Joanne, for doing this. And then go ahead and just talk about the 5 pillars for a bit. Then just share some concluding thoughts, and then just do some acknowledgement. So it's going to be a pretty short presentation, I think. I always say that, and I end up just talking because it's something I'm very passionate about. So I'm going to try and go ahead and get started. But before I do that, 1 second, maybe I should stop sharing and share my slide again. My apologies, because I feel like I am going to be moving my cursor quite a bit. Let me do this again. My apologies. Okay, great. Thank you. My apologies for that. Just to give some context to all of this, the content of this comment, which I think Joanne really wanted me to talk about a bit today, is really done from a space of collaboration, just like this session is kind of talking about. And it represents a team of members from the African Microbiome Special Interest Group and the History Africa Microbiome Task Force. In terms of the individuals in this group, it's a group of researchers that meet regularly to work on collaboratively building, I'm going to use a lot of collaboratively as well today, building a network of microbiome researchers to enhance the capacity of microbiome research on the African continent. We want to ensure that researchers are not working in silos where they could have synergistic efforts with similar projects be more impactful and really just move microbiome research on the continent forward. I mean, I listened to Joanne's opening remark just saying that, we want to accelerate microbiome research. Yes, this is something that I really want to do, and I'm really glad this meeting is happening to work towards that as well. So these researchers are in different stages of their microbiome research with varying skills and capacity levels, obviously, across their labs, and as such, are quite representative to an extent of the state of microbiome research on the African continent. The members span from North Africa, South Africa, Southern Africa, I should say, West Africa, and East Africa. So these comments kind of emanated from, obviously, a review of the state of microbiome research on the continent, and also from lived experiences and knowledge gained from the activities of the group, a major one, which is the African Microbiome Day. That's a virtual event that happens annually and has been happening since 2022, which is intended to really showcase microbiome research on the continent. We're not looking for like the fanciest microbiome project, but just saying, what are we doing? What are people doing? There are people working, in tiny silos, like we said. So just kind of like an opportunity to like, what are we doing? So based on all of these experiences, we kind of came up with the concept of, or rather proposed implementable pillars for equitable partnerships, which we know is really the basis of moving microbiome research on the continent forward. I'm also quite happy to note that a few of the labs and institutes present at this symposium, as well as the PASMO lab, are quite active participants in this effort, which makes me very excited. And the efforts that have been put together to make this in-person meeting possible. So congratulations again, Joanne and the team. So like I said, one thing is quite clear. Research in this day and age really works better rather collaboratively. Be it vertically, that's the global north, global south, or global south, global north, or horizontally, global south-south partnerships. Because at the end of the day, we are not just doing research for the sake of research, but as a means to an end, a very good end, we hope, that will improve the health outcomes of individuals and just really improve life outcomes of people living on the continent. So like I alluded to previously, we highlighted 5 pillars rather, which are by no means all encompassing, to be able to just, have equitable partnerships, equitable research done on the continent that can help us, accelerate or move microbiome research on the continent forward. So I'll spend some time on these 5 pillars and just look forward to discussions during the meeting, and hopefully the conversations continue beyond even this meeting, because I believe that a lot of these are quite relatable. And I actually think that Nicki has given quite a bit of my talk already from the introduction. So this is going to be very, very quick. So just to get started, I've just put the table you see in the paper if you have access to it. I kind of reshuffled the 5 pillars a bit. And again, this is just a proposed implementation framework. Nicki stressed down a bit about ethical and equitable partnerships, which is something I'm going to talk about as well. Not to sound like a broken record or not to sound too redundant, but this cannot be overemphasized because partnerships, are crucial. We realize that when we look at the authorships and everything else like we see that sometimes you find out that research has been done on the continent of Africa, but you look for the participants, the researchers on the continent on that paper, and you find them somewhere at the back or somewhere lost in the middle, or just some irrelevant, not very obvious position. Understandably, the contribution has to matter. That is correct. But now I'm saying that, it's not all bad. It's not all, negative. It's great, but we know that it can be better. And we realize that education is key. Just knowing how to do these things better. Some of the things we highlighted in this paper is that, when we are talking about research, collaborative research, let's start early. We form partnerships. We talk about the research from the start, from the get-go, and just form ideas from the get-go, involving local partners early. That way, it can also involve the community early, and that way, the priorities of the community will be highlighted in the research to avoid the so-called helicopter research we talk about all the time or read about all the time. So the idea is that when research partnerships are ethical and equitable, they work well, and they can be better sustained. And we realize that in microbiome research, we don't just want to do one-point collections anymore. Yes, we want to know who's there and what's there. We want to know, have some mechanistic insights. And sometimes we need longitudinal studies. Being able to have equitable partnerships that benefits both partners would allow for such long-term partnerships to exist. So I'm just touching on this briefly because I feel that there's going to be hopefully lots of time to talk about this more during the panel discussion, and also because I believe that Professor Host has also shared some of the things I would have loved to share, so I wouldn't bore you with that as well. So talking about ethical partnerships and just having some kind of input by the local researchers from the start leads me to the next point of local research leadership, which is really, in this context, African leadership. It's very crucial in ensuring, again, that the voices of the local population is heard, the community is heard, so that the priorities are aligned towards them. But not just that, to make sure that when all is said and done about the research, we know that, if it's a long-term research, the partners, the Global North or whatever other partners are living, knowing that they've empowered the African scientists to be able to carry out their own research eventually. We understand that it's a learning process. We understand there's a learning curve. We understand that we're at different stages of microbiome research, and so the learning is important, and the willing to learn both from the African side and from the Global North side is crucial. I'll give a practical example here, and I think this is something Professor Host has also talked about, touched on a bit. A very good example is the H3 Africa Consortium. The H3 Africa Consortium ran for about 10 years, and what this did was in genomics research. It helped both early career researchers and even more established researchers to just have more studies come from Africa, solid studies, impactful studies that even when you look in, you kind of look to PubMed to see what studies in genomics are going on in Africa today, or some very impactful studies, you'll be hard-pressed not to find an H3 affiliate PI associated with that study. This is something that started out, rather small, with a vision that we want to improve genomics research on the continent. We want to improve representation. And at this point, we're at a stage where I think, okay, well, I want to do the same thing for microbiome research. We have a good scaffold to learn from, and this is something that we are encouraging and we're hoping, comes through. And this is very critical in the microbiome research because we know that the microbiome research is more, it's easy, I wouldn't say easily translatable, but you see it. I mean, you take a certain kind of food, you see your microbiome is changing. We see all of these things and I realize that, well, if we have local leadership with local knowledge, local relationships with the local community, there's a very great chance of advancing and accelerating microbiome research, faster because of just that local knowledge. And the knowledge gained from our global partners and the interaction and everything being learned and being exchanged, rather, knowledge exchange will help this go further and will also enhance the careers of the African scientists, thereby also ensuring that we are more confident in our research because we have learned and we know that it works, and we are able to further mentor upcoming researchers to make sure that this is done in the right way. All of these pillars are kind of intertwined. So the next one we'll be talking about is the contextualized global research. What this really just means is that we are going to be doing research, obviously, as we're already doing. It's a work in progress, where the local voices become clearer and it benefits the population, like we said. So now with local leadership and this kind of equitable partnerships, you're hearing what the priorities of the communities are. You're more involved, more engaged in the community to actually have a sense of what is happening. So now we're doing research that is locally relevant, beneficial, but also very beneficial to the global population as well. It's informing the global knowledge base and also the scientific body on how some of this knowledge learned or gained from Africa can be utilized in a broader spectrum. Knowing fully well that, and when we talk about representation, we know that a lot of African microbiomes are yet to be unexplored. And knowing that there's a lot that the African microbiomes can contribute to the global. And in fact, every underrepresented microbiome can contribute to the global space. So this is one way of enhancing global research as well, just doing contextualized research that benefits the local population as well as the global space. So governmental involvement. So we know that the government is crucial, one, not just for the funding purposes, but in developing policies as well. And also just to create a conducive regulatory environment for microbiome research on the continent that will encourage the researchers to do the research properly and in a way that can have direct impact on policies. So on the researcher side, involving government is crucial. On the governmental side, creating that environment that works for the researchers to be able to effectively tackle public health issues with microbiome research in collaboration with the other fields. So I've put lastly here, and I know I've kind of like talked about the governmental involvement for the standardized microbiome protocols. My time is running really quickly. Is the standardized microbiome protocols. We realize that even slight variability in laboratory results can alter what we see in the microbiome. So it is kind of crucial, just going forward as we develop and increase microbiome research and build capacity on the continent, that we have certain standardized protocols. I know that this is also something that's being done on the global space as well, to ensure that what we are getting can be used in the future, even if it's for meta-analysis or similar collaborative studies with just using data, that we have a basis, set a good foundation to make sure that this is possible. And to this end, I'm just going to move on quickly now from this, that, like I said, standardization is of some protocols. Some protocols is an important element going forward, really, because we don't want to make everything look similar, but we have to make sure that certain things like data collection, that work well for the African setting is in place in kind of uniform. To this end, we also had another comment recently that just emphasized or just talked a bit more on the importance of standardized data collection and some of the strategies. And also the African Microbiome Special Interest Group and the H3A African Microbiome Task Force also created a tool to facilitate such collections across the continent. So it's readily available. Sorry, I've not put any QR codes, but these are readily available tools with some very good tutorial as well to help. This was done in collaboration with the H3A Bionet as well. So just some concluding thoughts. I know my time is fast spent. Just very quickly, I believe that regardless of the challenges, collaboration is key. We're working on it, and it's been improving gradually, steadily, and we know that we can always do better. The future is very bright for African microbiome research, and learning how to best implement frameworks to realize the vision for impactful microbiome research on the continent is a continuous and evolving effort. And I'm really excited to see all the initiatives ongoing to make sure that it comes to fruition. And I just want to encourage us to work together synergistically, despite the different initiatives. The underlying goal is the same, to move microbiome research forward on the continent and just have us more represented on a global scale. And I really do believe that together we can. And just on my final slide, to just say a big thank you to the organizers again for the opportunity to just come to about something I enjoyed very much. And also the African Microbiome Task Force, the African Microbiome Special Interest Group, and all the funders that kind of saw us through the periods of the H3 Africa Consortium, and as well to the institute that keeps me busy. This is the Brilliant for Molecular Bioscience. With that, I say thank you very much, and that is the end of my presentation.
Thank you very much, Ovokeraye. And then it gives me we'll come to questions at the end, like we have in the other sessions. Great pleasure to introduce Prof. Thulani Makhalanyane, who is a professor in the Department of Microbiology and the School of Data Science and Computational Thinking at Stellenbosch University and SERI in South Africa. His research has focused on understanding the ecology of microbial communities in extreme environments, as well as human gut microbiome in urban and rural South Africans. Thulani is editor-in-chief for the ISME Journal and senior editor at M Systems. He was elected to the International Board of ISME and was appointed director of the ISME Ambassador Program, which promotes microbiome outreach activities in over 100 countries at last count in 2018. He's one of South Africa's most vocal thought leaders on improving representation of microbiome datasets from Africa and developing better mechanisms to increase microbiome research in Africa. Welcome, Thulani. Thanks. Thank you. Over to you.
Thanks so much, Nicki, for the introduction and for your kind words. Thanks, Joanne. So it's the second time I'm in this auditorium. Most presentations in Cape Town, thanks to you. So today I'm going to talk a little bit about what you asked me to talk about, and I've got a subheading, which is what I will be talking about, which is the role of ethics in advancing global microbiome research. So Nicki's done such a good job, actually, just giving my talk before I got here. So I will try and say more or less the same things, but in my own style. And hopefully it seems like I've said something new. So I moved from University of Pretoria around almost a year ago now, where I've joined Stellenbosch University, where we've started the African Microbiome Research Group at Stellenbosch. So our interests vary quite widely. We focus on microbial diversity. It's a major theme of our studies. We focus on trying to understand evolutionary adaptation across a variety of different ecosystems. We're quite interested in understanding microbial interactions, and we do this using the One Health framework, with the understanding that microbiomes, as we've heard today, are the most important thing you've ever heard of. And that microbiomes underpin One Health. So you cannot get to a point where you understand animal health or human health without understanding the diversity, ecology, and evolution of microbial communities. My slide just keeps transitioning. So these are our thematic research areas. We're interested in African microbiome discovery. We've got a large ambit of our work, which is focused on training the next generation of microbiome scientists on the African continent. We're interested in establishing microbiome services and support through enabling a startup research culture within South Africa, but also widely within the African continent. We're interested in also coordinating microbiome policy and education. And our aim, which is our fourth thematic area, is to think about how we can strengthen regional and international networks focused on microbiomes. So we've got several different research areas. I won't talk about these to any great extent, but the main thing that you should see is that we're interested quite a bit in understanding how climate change affects African microbiomes, and we're really interested in establishing a large program focused on microbiomes in human health. So I'm going to talk about the underrepresentation of the Global South in microbiome research, and I'll use the case of African microbiomes as a case study, right? So Nicki, in a very nice way, said very strong statements, but because she's kind of prepared you, I want to really put my full support in terms of everything that you've said, but maybe add a little statement saying, if you're from the Global North, it's not that we don't like you. We really like you. But we do feel that the work that we do on the African continent ought to be led by African scholars for a variety of reasons, which I'll discuss. So this is a slide I got from a colleague of mine who is a plant scientist. He works on all sorts of disgusting stuff related to plants. But what he put up here is a slide that I've called the evolution of helicopter science, and it's an example of plant genomics. So what he tried to convey here is that initially, helicopter science started off from, people sending samples from Africa. It's evolved towards us actually sending DNA to other parts of the world. And it's now become more sophisticated in the sense that there's all these training, empowerment, and capacity development programs that are supposed to, in a way, promote research on the African continent. But what it ends up doing is that the intellectual skills and the real value in terms of plant genomics ends up on the top end of the slide, which is where really the ideas are conceived, and that's really the value proposition within this field. So there is something quite similar when it comes to microbiome research on the African continent, and we also essentially doing the same, right? It doesn't mean that we don't value the corporation, and my timer hasn't started, Nicki, so I can take as long as I want. But it means, that's not what that timer says. So as one of the main reason is this plot that a few people mentioned, which is the increase in terms of the demographics. And what you will see is that Africa is projected to increase quite considerably. We've got a young population, but what this implies is that this population of young people are potentially a large genetic reservoir, intellectual reservoir, where important questions involving microbiome research on the African continent can be led by these individuals. We need to figure out, as our population increases, how we can tap into this capital to solve our problems on this continent. And this needs to be driven by us. So this is a slide that shows kind of the discrepancy in terms of the current outputs related to microbiome research. If you look at the top slide, when you put in microbiome, you see over 88,000 publications. When you put in microbiome in Africa, which is as a proxy for papers that are published from the continent, it's a dismal 446, right? And you can see that when you look at global outputs, they span a variety of research fields and over 30 research fields. And that when you look at comparable data for Africa, it only spans 11 research fields. What this shows me is that there's tremendous potential in expanding microbiome research across a variety of other research areas. And this, for our growing population, has got tremendous population in terms of creating jobs and also solving problems that are related towards the wide genetic diversity that we have on this continent. So this is a figure that we put together for a review that we published. And this just kind of underlays and shows you visually the problem that we're sitting with. When you look at, so these yellow and green dots show the data that we retrieve from the NCBI. And you can see that when you look at all of the data sets that are currently available, there is a big gap when it comes to microbiome data sets on the African continent. And when you look at the type of data sets that exist, they're mostly related to host-associated microbiomes, because in a lot of studies that are conducted in other parts of the world, African individuals are usually used as an ancient sort of model to kind of compare prehistoric diets. That's not really questions that are probably of relevance to Africans, and maybe they do not necessarily solve African problems. So it also kind of makes a point of why we need to kind of conceive our own research questions and take the lead in terms of addressing these. So we've said and we proposed that we want to have a large genomic sequencing project called the African Microbiome Project, where we want to sequence 10 million samples in the next 10 years. So we've also sort of come up with a roadmap in terms of how we want to expand microbiome research on the African continent. We see the coordination of large research networks on the continent as being central to this. We see policy interventions, the sort of policy interventions that Jacques has just outlined, as being also a second key element to this. And we see also how we can share infrastructure as being core towards advancing microbiome research on the African continent. So it's quite fashionable on this continent that everyone wants their own sequencer. But what we're trying to move and promote is how we can essentially share the available research infrastructure and focus more on solving the research questions rather than trying to acquire new bits of machinery. Right? The reps in the room will not like this at all. So this is our first meeting that we have in Cote d'Ivoire last year, where we met. We had researchers from around 12 different African countries, and we plan to meet again next year, probably in Ethiopia, where we want to continue cementing this network of African microbiome researchers. So we've articulated these views in 2 different articles. The first one, African Microbiomes Matter, is available, and Jacques has gone into detail about the sort of proposed framework that will allow us to promote more equitable partnerships. So when we talk about how we define ethics in microbiome research, we wrote from an article a few years ago. It's a hard concept to try and to define ethics. But what we arrived at is that microbiome ethics relates to several recognized fields of ethics, including bioethics, environmental ethics, and food ethics. and that it's underpinned by four cornerstone principles of ethics. And that is to do good, don't harm, respect, and act justly. So what does this actually mean when it comes to the research that we do? We've sort of, if you read this paper, gone through what this might imply across different areas of microbiome research, and what it actually implies in terms of being a microbiome researcher that acts equitably. So there are 5 key ethical considerations, and I won't talk about them in great detail. But the first that Nicki has also touched on is to think about how we can have equitable data sharing, to think about how we can have global research collaborations that are not necessarily biased to the global north, but that are led by researchers in the global south. We need to think about how we can have benefit sharing, have proper environmental stewardship of African ecosystems. The last one that I'll talk a little bit in detail is that often we think, and I've heard it quite a lot in this room today, where people talk about consent and privacy. I feel that in our own human microbiome work, we probably do not do justice towards informed consent, in a sense that the type of samples that we get from people, we know through metagenomics and other tools, have the possibility of having these data being retrieved on the NCBI by startup companies that commercialize this research and produce millions of dollars. And I don't really think when we go to communities and have people sign documents that they understand what the power is of the data or the samples that they're giving away. We all need to do a little bit better in terms of really explaining what consent and privacy actually means within our field. And just to conclude, so I've touched on a few things, and hopefully you grabbed one or two things, but we urgently need to expand microbiome research in the global south. We need to reduce some of the barriers that impede research on microbiomes in the global south. We need to continue collaborations with researchers in the global north, but these collaborations ought to be equitable in a sense of that should not be one-sided in terms of the research that we do. With that, I'll bring my presentation to a close, and I'm exactly on time no matter what your watch says. So thanks.
Thanks, Thulani, for a great talk. Okay, timekeepers, you meant to start it when they start talking. So it gives me great pleasure to introduce our final speaker in this session. Professor Jacques Ravel leads one of the globally recognized research programs applying genomics and ecological principles to study the vaginal microbiome and its role in women's health. His work has pioneered live biotherapeutic treatments for conditions like bacterial vaginosis and urinary tract infections, now being tested in clinical trials in the US and Africa. Prof. Ravel is the founding editor-in-chief of Microbiome and an associate editor at MBio. Prof. Ravel.
Okay, well, thank you very much to Joanne for inviting me, and also thank you for giving me a topic that I felt somewhat, outside my comfort zone, but I'll do my best. And following two fantastic speakers, I feel like I'm going to be somewhat redundant, but I think there's going to be new bits of information. So I think we probably didn't show this paper, so that's not redundant. But the issue with human microbiota research in Africa is it's been there for a long time, and this paper was published in Microbiome, the journal I'm editor-in-chief, and nothing to do why I present this one. But and this paper actually was one of the first to really highlight the issues that are associated. So, for example, they looked at a lot of studies and they mapped them around Africa, and only 33 of the 54 African countries were represented in 168 studies that were published at the time. This was in 2021, and primarily from some of the a few countries. Also, the founders were mostly American or European institutions. So already some of those problems were highlighted. Some of the problems also, Voika actually did a great job at talking about it. But if I want to add some of those that she mentioned, the first and last author of most studies were not affiliated with African institution, which was a big problem. But another one that I found kind of interesting is that among study with intercontinental collaborator, the USA was the most common collaborator, but there was very few intracontinental collaboration. And I think that's a little bit a major issue as well. It seems like there was a lot of one-on-one collaboration with outside of Africa, but this network within Africa that was talked about earlier is absolutely needed. And that network can work with other partner outside of Africa. But without that network, there's no power in Africa. So but there's definitely, in conclusion, the need for local leadership, capacity expansion, intracontinental collaboration, local government investment in Africa. And this was, I'm not going to talk about it, but this is what Volker just talked about. So I think that we need to build, and Africa needs to build, very large a microbiome collaborative network. Not just a few lab working on this topic. And I think this brings a lot of strength. I always think that diversity in numbers is actually a strength. And so if you have large microbiome collaborative network, you should include as many collaborators as possible to make equitability not only across the continent, but also within continent. And I think that's important. If a lot of partners are involved, a lot of partners benefit from being involved and access to knowledge and data and so on. So I think inclusion is very important across the continent. Of course, diversity and ethnicity and ancestry will definitely enhance that collaborative partnership and provide really important information, not only for at the local level, but also at the global level. And those networks should really focus on enhancing harmonization. And I'm going to talk a little bit more about this because I care a lot about this, because without harmonization, we can't actually share data. We can't even know and find the information that we need about studies that exist out there. And I really like that initiative that you presented, and I think it's going to be fantastic. Data doesn't have to be shared right away. Data can be just findable, and to be findable, it needs to be interoperational. And to be interoperational, it needs to use common language. And I'll try to discuss this a little later. And so I think that ultimately, what we want is scientific finding to be global and local. And I think that's what large microbiome collaborative network will afford. Leadership should be global and local. So we should have African leaders that are global leader. Okay. And of course, as such, will also be local leader. Ownership should be global and local, and the output of those networks will be global and local, and the benefit of it will be global and local. So I think there's major benefit to have large microbiome collaborative network. I think this is something that's very important. So what are the opportunities through those large collaborative network? And I think they address a lot of the issues that were brought up before. So I think they will definitely enable local leadership, okay, by including a lot of people. And also people will be able to bring cultural sensitivity. You would not have just like you have one, the model is the global north kind of dominate the global south, but we don't want one African partner to dominate another one. We want equality also within the network. And I think that by bringing multiple cultural and ethical viewpoint, all this will be considered in building this network. So I think this is another important thing. And of course, policy and advocacy could be leverage, could happen through those large collaborative networks across the region and the continent. So those are 3 points. There's 3 others I'd like to make. Of course, capacity building and training. So I really like the talk earlier because it seems like a lot of the capacity building is as we think of it, is like you said, America, Europe, or Asia invest in Africa. But it seems like now Africa is investing in Africa, and I think this is what's needed. And so capacity building can happen not only across continent but also within continent, because there's also inequality within the continent. Global data sharing is also very important. I mean, to make important discovery, whether they are at the local level or they are at the global level, global data sharing is important, and I'll talk about this, as well as harmonization of laboratory practices, analytical protocol, but also what I'm very passionate about is the use of common data descriptor and open access tool development that can be enabled through those kind of initiatives. So, but of course, there's a lot of challenges in implementing and making those opportunity a reality. There is definitely resource disparity, even not just between continent, but within the continent, and that needs to be addressed. And there might be some way to bypass this limitation. Some of the tool, if you can be creative and in the way you access analytical tool, for example, you can bypass the need for large servers. There are ways to get access to those kind of infrastructure without building locally. But of course, ultimately, we need to push for global African investment that needs to happen. Data ownership and intellectual property is always a big thing, and it obviously should remain at the local level. Even though it might benefit ultimately the global level, at the global level, it should be owned and at the local level. And the major issue, of course, in terms of challenge is long-term commitment. There's been a lot of investment, but they could be short-term and then they terminated for no reason. And then, you're left with nothing to build on. And so that could be another issue. And those commitments should also happen within the continent. So databases is very important, and representativeness in databases. So in the human vaginal microbiome space, we had built this catalog, which is a comprehensive catalog, a gene catalog of all the genes that you find in the human vagina. That was Virgo 1, and it was basically a US-based catalog. Okay, that's where the data that came from. And so now we just built Virgo 2 with some funding from the Gates Foundation, and we've tried to expand a little bit the global representation of it. But there's still a lot of work to be done, and even though we think it's highly representative, we have data set from a lot of parts of the world, there's still, like, if you look at those accumulation curve, there's still a lot of diversity to be discovered. And we really need to work together to make this a more comprehensive database and tool that can be used everywhere. And so we need more global data, and I think we should all work together to do this. So Virgo 2 is not just a database, but it also comes with a whole set of analytical tool that can be used to analyze metagenomic, transcriptomics, and metaproteomic data set, which are now becoming quite common in microbiome science. So we can all work together to make this better. And of course, it should be totally open access and fully available. All these databases, they become a useful tool for everybody. But if people don't have access to it, it's not very useful. So this database is open. It comes not just the database, but even the data that was used to generate the database is available for everybody. And I know a lot of people have downloaded those quite large data set, but they were able to download it and leverage them. So the other thing is standardization and acquisition of data and data description. So of course, we talked about standardization of workflows. There's absolutely no question. But I think one thing to think about is, like, standardization doesn't mean you have to do what the Global North is doing. Standardization means to come up with a compromise so things can happen equally at both places. So it's a two-way street, okay? It's a two-way process. And it needs to be considerate of local capabilities in one place and the others. Otherwise, you don't have a harmonization. and then it only works in one place. Of course, it should go all the way to analytical workflow. And remember, harmonization doesn't mean complete standardization. Everything has to be the same. If you consider local capabilities, as long as they are validated, the process can be different. And I think that's important. A lot of people think like, oh, I have to use this kit because that's the kit they use over there. No, it doesn't have to be that way. Harmonization is not standardization. It's not just the same process. It just means you get the same results. But you can get there in a different way. But if you get the same kind of result, that's comparable. So that's very important to consider. Analytical workflow also is important to standardize, obviously, but there's also this issue of local computational capability versus cloud-based access. And I think the cloud is, to me, is a fantastic tool because it enables people with just an internet connection, which is becoming standard pretty much everywhere. Even on phone, you could actually access the cloud and run processes and do quite complex data analysis. So I think that's a tool that could be definitely leveraged. And of course, data description. And I feel very passionate about data description because we can't collaborate if we don't talk the same language. And in terms of same language, it's how we describe data. Okay, if I call a set of data one term and so you call it a different term, we'll never get to meet together. So I think we need to use and develop common languages to describe everything from participants, sample, processes, any kind of metadata, data type. Data type is very important. I mean, just like people call 60ness RNA, gene sequencing, genome amplicon. There's like so many ways to say that same thing. But in databases, it creates problem. We can't find data that were generated the same way. We can't find data that should be comparable. Okay, so data description is extremely important. And so we should really use ontology that afford this easy cross-referencing and maximize the value of global database. And those common ontology, when you have large network, you can all come up with a set of language descriptors that you can use. And those descriptors, because you have this large network, you have power and everybody's using it. So now you have this large network of people that all talk the same language. If you develop this locally and somebody develop another one and another one, then those languages are not going to talk together. So having those larger networks really help in agreeing on those common descriptors. And of course, we have those principles, which is the FAIR principle, findable, accessible, interpretable, and reusable, that can describe data set. And so without those standard languages, we can't actually achieve the FAIR principle. And some of the ontology that we've been using, they're really common ontology that's used across the field of data science. You have the EDM standard for any kind of data type or even format. Like you generate a sequencing file or analytical file, it has a format. Even if it's an Excel file, it has a format. And you have to have one descriptor that is very important that says, this is the file I generated, and it uses this particular descriptor. And everybody use that descriptor, everybody will find that data, whether it's a FASTQ file for sequencing and so on. But it's very important to have very good description of those. Also, OBI, which is Ontology for Biomedical Investigation, that represents everything you're going to do with the sample, the assay, the instrumentation, the reagent used. All this is also very important. People don't really think about describing this as opposed to describing the sample the data come from, okay? And of course, also anatomy is also very important. And so Oduaran is another ontology that you can use to represent a variety of anatomical criteria, structure, function, even developmental lineages. So it's very important. And of course, we all talk microbiome, we talk bacteria. I mean, taxonomy is essential. If you can't even agree on how to name things, granted, things change all the time, but if you can't agree on how to name things, then you'll never be able to collaborate really very well in those things. So how can we improve those global database? So we need to expand global and local insights. So we really need to add more diversity into those databases, and integrate a lot of different ancestries into the database. This will improve global and local health outcome. Obviously, the more diverse you have, the more refined you will be in your analysis in order to have an impact at the local level. And of course, you'll be able to do more transformative research and able to actually leverage this in more practical application. So one thing also that's very important is capacity building. And I wanted to talk about this because I participated in a workshop that the Santé program, and this is an advertisement for the next one. And you can see it's funded also by the Gates Foundation, the Santé program, Harvard, and others. And I participated in this workshop, and I got to meet a lot of those participants. And to me, it was kind of inspiring for several levels. So first of all, it's providing training in critical computational analytical skills. So, and to me, this is kind of the last link. Okay. And I got to talk to a lot of the participants there. And what I was amazed is a lot of them were actually, they started as like technician in laboratories. Okay. And they were like, they used to be the one that just collect the sample and ship them to Europe or America. And then they started processing them a little bit more. They did some analysis, but then the data was being shipped over to, again, Europe or North America. And now with this, they see this as the last link because now the data is staying locally and they can actually perform the analysis themselves. Not only it empowers them because they grow in their professional aspiration, they're becoming more computational, which gives them a lot of advantages and benefit, but they also now able to process, to go from the beginning to the end. They don't need anymore to ship anything. And so this is the last bit of this empowerment. So I call it mentoring to independence. I think this is really what this was. And it was also very nice because we saw that some of those trainees were so good that actually Doug picked some of them and Joseph, and some of them are going to be actually teaching the next few workshops. So they became actually the teachers and the owners of those workshops. And I think this was something that I think was really nice. And of course, the workshop is leveraging cloud resources, which people left and they were still empowered to continue using and what they learned because they had access to the cloud. So the last thing is I wanted to talk about is because Joanne asked me to mention a little bit about translation and how you can understanding more global distribution helps you a little bit in trying to leverage this into something more practical. Like, for example, for live biotherapeutic product development and for vaginal LBPs. So local strain for local use. This has always been the big issue. Should you use local isolates locally or global isolate would work? Well, in a vaginal microbiome, it's actually quite interesting because what we see is that, for Lactobacillus crispatus, which is the species that's used in the product that Doug even mentioned earlier, you can see that some lineages are very unique to certain continents. That means those strains are like endemic to certain countries, that you don't see them anywhere but in those regions. But there's also strains that we call cosmopolitan, meaning that there are strains we find everywhere, and there's both. So the answer, can we actually answer this? No, not yet. We're going to have to do the work, and people in Africa is going to have to use African strain and test them, and we'll be able to see if those cosmopolitan strain from Africa actually are needed to potentially support those endemic strain. So there's a lot of things we can learn, but we can't generate that information if we don't generate the diversity, if we don't exploit that, understand that diversity that exists. So with that, I just want to conclude that together we should leverage this diversity to establish those global programs that's not across continent, but also within continent, that are equitable and empower global and local scientists and communities equally across and within this continent. We could continue to increase our global representativeness in microbiome database, because I think that it's going to enable translation. But in doing this, we really need to start talking exactly the same language and agree on how to describe the data. And of course, continue training to empower this idea of mentoring to independence, and go all the way, all the way to publication and translation. This is not just necessary to do the analysis, but you need to even take it further. Leverage that information, be creative, start thinking, not just replicating, and leveraging the microbiome for discovery, and of course, develop African-led intervention and clinical trial in the microbiome space to enable business opportunities. So with that, I'd be happy to answer questions.
Thanks so much, Prof. Ravel, for a great talk. Can I ask Thulani to join us up front, and we can get Voka on the screen to join our panel. And once she's up there, then we'll be ready for questions. So get your questions ready and lined up. I've got a few. I promise they're easy. Yes, Joanne. And please remember to just say who, obviously Joanne doesn't have to, but everyone else, please just remember to say who you are and where you are coming from, country or institution, whatever. And then we've got Scott. Okay, Joanne, over to you.
So I think I'm waiting for the mic. Let me just speak to this way.
I will do. Oh, there we are.
Great.
So I've actually got a comment to both of you, actually, because as I was with the co-chairs for this meeting, working through the lineup and what we wanted to discuss and what we want to create, and this whole meeting is about this call to action to partner and collaborate, not just in South Africa, but we literally, the work coming from yourself, Thulani, and from Voka's paper that you co-authored with her, it is the framework. So maybe my question to you, if I had a question, what does the future look like? Where are you going with this thinking? What are your thoughts about hub and spoke model? And then before somebody else gets to ask a question, I just wanted to say to Jacques that, others outside of the vaginal microbiome field may not know that this is pioneer for a lot of the stuff we do and a lot of the standardization. But I just wanted to also say that what you represent is an enabling partner. And on VMRC for Africa, the one thing that's occurred to me through this Coletta Zuma Fellowship is each one of us have been selected to be part of this, most of us have a really powerful enabling partner that allows us to elevate what we do. And it's quite a rare thing. So I just wanted to thank you for the opportunity.
Thank you.
But Thulani, back to you.
All right. So I think it's on. So yeah, you asked what my thoughts are. I have many thoughts. But the first, I think, maybe directly linked to this, one thing that I'm quite passionate about now is thinking about how we move beyond what we've done before, which is just short-term training courses, and actually translate that to producing actual research. So I think there is a good value in bringing people together for 2 weeks and training them. But I also wonder about the ethics of sending them back to their home environments where they don't have the facilities and the capabilities to undertake that research. So moving forward, I think what we need to think about is how we enable that whole ecosystem to allow people to undertake the research and think long term. So something that could work for us at institutions is to maybe think about how we can not have short-term fellowships, but translate these into maybe sandwiched PhD programs where you pair people with specific mentors, and then they can work on issues that are directly applicable towards their context, for example. So that's just one example. But it's really trying to think about how we combine all of the different efforts, but start to have real traction in what we do.
Would you like to also chip in, Jacques?
No, I mean, I completely agree. There's danger to train people and then send them back to and be then that maybe the discussion about flight risk. I mean, they just want to leave, and you're not building anything locally. I think this is definitely a very important issue to address.
Voke, I can see you online, although you're not on the screen. So if you would like to add something, will you also nod or raise your hand? And I can certainly see you to bring you in. Yes, carry on. Go ahead.
Great. Really, really fascinating talks. I was just going to say that in addition to Thulani's point, on just having long-term trainings that are sustained, I think that in terms of Joanne's question as to the next step, I think we all talked about this a bit, the importance for the networks. I think one way to just get this networks going is like a meeting like today, and then just having like more frequent, thought processes and meetings to just how are we doing? Let's check the pulse every night. How are we doing? What is possible? How can we work together? Like I said, there are so many initiatives. Are we doing things like, in a divided form, or are we doing things together? Because if there are several little different initiatives going on, that might take away from the entire process, because the entire goal is to let's do this and bring, increase representation. We want better health outcomes. We want to be able to do this precision method everybody's talking about and contribute a microbiome bit to it. Just like that pulse check frequently. And just to say, okay, we can do this together. We can work on this together. We don't have to do this in little bits. Yeah, that's my take. Thank you.
Great. I'm going to hand over to Scott, who had a question.
Thanks. I've got two questions. The first one is to all the speakers. I think the one additional aspect that I think we need to talk about is we spoke about in the session about the relationships that scientists have with each other. But I think the other important party are the communities and the participants in our research projects. And I think there's both an ethical imperative to involve them throughout the process, and I think also a very self-interested reason to do it. We've done a pilot study now of giving personalized feedback of research results to participants. And what's clear is that participants really want that. So I think then there's an ethical issue we need to think about. But we also know that if we want to go back and work in those communities, or we want to recruit those participants again, this is something that we need to do. The second question, I think, is primarily for Jacques, although I'd be interested in what others have to say. In the human genomics community in H3 Africa, the policy of data sharing was that all the data had to go into the European Genome Archive. And then there was a biospecimens and data access committee that then gave people access. And one of the conditions was that for the first 3 years, I think, is that if you wanted access to the data, you had to have an African partner. It didn't have to be the people who put the data there, but it had to be an African partner. And I wondered what your perspective is on having this in microbiome research, because in a way with non-human data, we don't have the same culture. It goes into ENA or SRA, and it's considered to be freely available. But I think there are real issues around equity about just putting the data into public repositories.
So there's different level of sharing. So obviously, I'm very passionate as a journal editor that if let's say you write a publication with data that you generated on microbiome, whether it's in Africa, in Europe, or wherever, and you do some analysis and you publish it, whatever you use for that analysis to be reproducible, the data needs to be available to everybody else to be able to reproduce. And I feel like this is essential for your credibility as an author, and for the field to maintain its own credibility. Because there's a big danger in the field of microbiome, which is lack of reproducibility. And so if data is available for reproducibility post-publication, then it's a win for everybody. So this is post-publication. And I think that's very important. But I'm not advocating at all to put data in public databases prior to publication. What I'm advocating is to put, and that's why I really like this idea, it's put descriptors, data descriptors that are common, that is searchable, that is very accessible, where we can actually find that data, and then you can actually contact whoever's generated that data and strike a partnership with us. So I don't think data needs to be publicly available prior to publication. So I think that once you have your publication, there's potentially less risk to be scooped.
No, I think so. If I can respond, Nicki, I think the issue often is the case is that the data can be used for additional research. So the need for the local group to have some time, because we don't have the same resources to be able to compete as with top labs in Europe and North America. So the database, the access committee, if someone said, I want to reproduce your work, then the data access committee can give them access to the data for the purpose of reproducing the work. But if they're now wanting the data to do additional research, then I personally think it's reasonable to ask them to have an African partner to do that research.
Absolutely. And I think that like free access doesn't have to be like, if you go to like even places like the equivalent of like dbGaP, where it's still publicly available, but it's under controlled access, you can put a limitation on any limitation you want, which will be, okay, if we will give you access to this data, but you need an African partner. I think it makes a lot of sense, and it's your prerogative to do that. And I think I would support something like this.
And if I can just comment, I know as a moderator, I'm not supposed to have too much of an opinion, but those of you who know me know I'm horribly opinionated. But I think there's also some kind of curation needed before data are available openly. And one of the things is, of course, human sequence that contaminates, human genome sequence that contaminates sequencing from a variety of resources. We're looking just at wastewater sequencing. And of course, if you're a pathogen specialist looking at wastewater sequencing, all you see are pathogen DNA sequences that feel ethically not very charged, but mixed in there's a whole lot of other stuff. And I think that some kind of curation, some kind of oversight, it's actually essential to protect participants, because of course around every microbiome there's probably wrapped a person. Yeah,
it's actually even worse in our field of vaginal microbiome. If you sequence the metagenome, not amplicon, 90%, sometimes 98% of the sequences are human. And we need to remove them all before the microbial part is released. But yeah, and often I know at NCBI, they also do their own validation to make sure there's no human sequence in those data sets.
Yeah. Questions. So I don't know who was the first out of you two, but I promised to honor you both. Is that Val?
Yeah, thanks again. It's Val Mizrahi from UCT. One of the themes that I've heard today coming through loud and clear is this question of equity and fairness in research. And we are here in Cape Town. And I just want to remind people who are not familiar with the Cape Town Statement on Equity and Fairness in Research that was published in Nature about 2 years ago. And I was really reminded by Thulani's talk, where he framed these questions in an ethical context. But what the Cape Town Statement came out of was a conference on research integrity. And the key about equity and fairness in research is that it's now framed in the context of integrity of research. This is not a negotiable anymore, as far as I'm concerned. And if people are not familiar with this, I would urge you to look for a paper in Nature. It was published about a year ago by Lynn Horn, is the first author. It offers a set of guidelines for people from privileged environments and less well-resourced environments to really think very carefully before they enter into something that is even called a partnership. So that's just a plug for the Cape Town Statement, and I thought it was absolutely appropriate just to remind people of that, just because it came from here, from a study that was led really by ethicists and people working in research environments and research support systems. And one of the key differentiators is we often think about equity as between individuals. What they highlighted is that enormous inequities exist between institutions at the meso level. And I think we here in South Africa need to be very, very humble when we participate in partnerships with colleagues in other African countries who might not have the resources that we've got. Thank you.
Thanks, Phil. Thanks.
Sarah Gitome from Kemri, Kenya. Thanks to the three speakers for the great talks. I think today I've heard a lot about the need, and I totally agree on the need for sharing data from our research as a way to promote equity and also sharing lessons. What I'm wondering about is what extent do we leverage other, perhaps less research kind of networks, for instance, routine healthcare services, people coming in for routine healthcare services, or even HDSSs, that's health demographic surveillance systems, as a way to still collect this African microbiomes data. And can that be a resource for attracting funding to build local infrastructure, local expertise? We analyze them here. We understand the evolution of diseases. We understand how the microbiomes are changing over the life course of different kinds of people.
Thulani?
Sure, I'll pick a stab at it. I think it's an absolutely great idea to leverage existing infrastructure. But the thing that should underpin this is the research questions that we have, right? And if these are research questions that all the participants that are donating their samples are well aware of, then of course we should absolutely leverage that infrastructure.
Do you want to add something, Jacques?
Yeah, I'm not quite familiar with those medical record infrastructure in Africa. It's like, do you guys have like global, at least in like just in South Africa or in Kenya? Is that for the country?
So I can talk to South Africa and then maybe you can talk to Kenya. So I worked for about 4 years full-time at the Department of Health in the Western Cape. So health service delivery here happens at a provincial level, and each province has a different system. Our Western Cape province has a health data exchange that does pull in all the records from all the digital platforms, primary healthcare, hospital level, from labs, from pharmacy, from emergency services, etc., from registers, etc., and that comes in daily and is linked to a patient master index. So we do have a full longitudinal medical record here in the Western Cape for every health service client from the public service. So it's about 7.5 million current patients. But that's not the case in other parts of South Africa. So maybe you can speak to the Kenyan situation. Yeah.
Yeah, so in Kenya, we don't yet have those centralized kind of like electronic medical record systems. You do have some facilities which are piloting, maybe in like different counties. You might find that there's a county that's trying to pilot those systems for their facilities, but it's not, national. But we do have a number of HDSSs in different regions that follow people over a long period, several years, collecting various indicators and tracking various diseases.
So Voke has been waiting to comment. Do you mind? Oh, okay.
Thank you. So I'll just
the whole discussion period, all the online all the online people can only see her face and not the audio, not
Oh, okay.
so that's why. So I've just said to
Yes, please, Voke, over to you.
Okay, thank you. And Scott, not to put you on the spot on this one, but I'm going to defer to you a bit on this. So speaking of HDSSs and leveraging, the facilities that exist, I believe that something can always be worked out because we do have not to toot our own horns, we do have a study in South Africa actually that has effectively utilized HDSSs to carry out microbiome research, and I believe it has worked out very, very well because it has such an amount, like you said, Sarah, an enormous amount of data, not just microbiome data, but data that could be widely applicable and just contribute to the robustness of the microbiome study. I don't know if Scott, Prof. Hazelus wants to say something, one or two things about this.
Thanks, Sophie.
If I can just, for those who don't know, an HDSS is a health demographic surveillance site, and these are very large longitudinal cohorts that are embedded in the community and follow up consenting participant literally over years and years. And they were very instrumental in the COVID research because they were very agile at tracking in participants for whom they already had a very good record, how they were doing with COVID-19. That's just to put you in context. I've got one question from the back. Is it Moses? I'm peering into the dark. It's dark and I'm not wearing my specs, so apologies.
Thank you very much for the speakers and a very beautiful session. There are two questions I wanted to ask. First, through the reading, I don't know, I've been battling with this. Do we have an African equivalent term for microbiome in any of the dialects or any of the languages? I think that's an important element for community engagement. And this brings me to the second question. In the aspect of One Health approach, we have the environmental microbiome, human microbiome, and the animal microbiome. How do we leverage this particular concept and make sure that the communities are well engaged? Because I know like the uptake of vaccine has been poor because there have been so many myths around vaccines. When you get microbiome to soil health, for example, and want to use biocontrols, biofertilizers, there could also be similar kind of resistance. So how do we ensure that we have communities engaged from the word go and take this concept as one of their own? Thank you very much.
Can we start with Thulani? I feel like this question is initially yours, and then I've also just been told that's the end of questions. So if anyone has something burning, then maybe we can just squeeze it in, and then we'll cut it off. Is it a burning question?
It's actually
we can
quite
let
related.
them answer and then come to you. So maybe we can keep it a little bit short, if you don't mind. Thanks.
You can go for it if you want.
I think you should go first and answer Moses' question.
Do we have an African equivalent? I'm about to find out because we're having one of our ethics approval translated into isiXhosa. But I'm likely to guess it's not a one-word sentence. It's more a descriptor, which is why the translation costs a lot of money for isiXhosa.
Okay, great. And the One Health question of microbiomes from the different categories and the different ecosystems that Moses asked about, how do we leverage the fact that there will be sort of veterinary microbiomes, animal microbiomes, plant microbiomes, human microbiomes? How do we bring these all together to talk to each other? And it's an open question. Okay, I'm watching you for a wave. If you want to chip in, Jacques, maybe you have a thought around it.
I mean, it seems like, from what Scott was saying, it's like participants want to know when they contribute to studies. And I think that it's going to be the same from people who might be affected by whatever intervention or that is developed, whether it's for animal, whether it's in agriculture or it's in human health. I think information is power, and I think that it's really important to bring that. How do you do it? That's a challenge, and I think people need to think about it.
Thank you. So, okay, you have your hand up. Quick response.
I'll just listen very quickly.
Oh, wait, we need sound.
Can you hear me?
We can't hear you.
Okay, I'm not quite sure, but
now we can hear you.
Okay, great. So I was just going to say that in terms of the translation of the microbiome, I think we've had Scott talked about the community engagement being done, and some of this was translated into the local languages. I know that some stuff was translated into Swahili, and that's currently ongoing right now at APHRC. So there's been some translations, and hopefully we'll find out about that soon in the more public space. But there's been some translations, yeah. Great, thank you.
Thank you. Do you want to put forward a last comment?
Yeah, I mean, I think we should think about this, and I know we don't have time to talk about it, but I am worried about going back to communities and giving results of our research when sometimes we don't even understand what the implications of those findings are. For example, we told women for a long time with HIV not to breastfeed, and then all of a sudden we realized that breastfeeding was better than formula feeding in some settings, and it's been really, really hard to overcome the previous messages we've given them, and with vaginal microbiome as well. Like, for example, we did a study where we looked at whether or not vaginal insertional practices altered the vaginal microbiome, and we didn't find that they did. But the number of women using VIPs was fairly small. So I don't want to go back and tell women, go ahead, use your VIPs.
I just think we have to be careful and we need to discuss it a little bit further.
I think that's a great point to end on, and to thank, I'm sure everybody agrees with you, so I'm not even giving them an opportunity to disagree. If that's all right with the speakers, because we've run out of time. So I'd like everyone to join me in thanking our speakers, Ovokeraye and Thulani and Jacques, for a great session. Thanks very much.
And thanks to Nicki for kindly agreeing to chair the session. So the last piece I just wanted, and this is way more important than this really important discussion going on, but we've got this opportunity to get a group photo, and I don't want to miss that opportunity. So I'm going to invite everyone to go upstairs for a really important group photo. And following on from that, we'll have the official opening of the Microbial Interactions Lab. And I know everyone's had an extremely long night, a long day, but I just wanted to thank my co-chair, Lenine Liebenberg, and Stellenbosch University in Surrey, who have sponsored the wine from tonight, because Stellenbosch University has their own wine farm. So I just wanted to say thank you for that. But if we can just go upstairs to get a group photo.